Hi, everyone. We've been doing a lot of work as it relates to the notion of traits are transmitted through monohybrid, dihybrid crosses. All those things are there. But we don't inherit genes, we don't inherit traits, we don't inherit alleles per se. What we do inherit, as we know now, are packages of genes, packages of genetic code, which are contained on our chromosomes, which are actually passed along in our gametes. Now, this was a foreign concept for a long time. This is actually covered off in your text. But if you go back in time, in fact, way back to 1902, there were two scientists, a gentleman by the name of Walter Sutton and also Teodoro Bovieri. They were looking at meiosis and they put together a few things. They noticed that the chromosomes behave in a manner similar to the way genes behaved, as Mendel described. To that end, these two individuals made three key observations: that one, chromosomes occur in pairs and these pairs segregate during meiosis. Secondly, chromosomes align independently of each other along the equator of a cell during meiosis. And three, that each gamete will only receive a single chromosome from each pair, very much mirroring the findings that Mendel arrived at. So let's take a look.
Let's give ourselves a reminder of how important Gregor Mendel's work was in 1866, that he published in 1866. And it's important to realize that it wasn't like immediately in 1866 or 1867 the whole world changed and everyone said, oh, Gregor Mendel figured it all out. Like a lot of times in science, the big discoveries, the ones that really change people's thinkings, aren't really taken that seriously at first. And actually Mendel's work, a lot of people either didn't take it seriously or kind of ignored it in 1866. And it wasn't until the early 1900s that people rediscovered his work and realized, wait, there's something very, very powerful here. And we might be able to connect it to things that we're actually observing inside of cells. But let's just remind ourselves about Mendel's work. So for most of human history, we've recognized probably that, okay, it looks like animals, or not just animals, any type of living creature seems to pass on traits to their offspring. I could look at you and I'd say, well, your hair is kind of like your dad's and your eyes are kind of like your mom's. Maybe your nose looks like something in between. You walk a little bit like your uncle. So we've always recognized that we pass on traits to our offspring, but we didn't really have a rigorous way of thinking about it. And we definitely didn't have any way to make predictions that were testable based on those traits. And that's what Mendel gave us. He said, well, look, I'm observing, and he did this with pea plants, I observe these heritable factors. And there might be heritable factors on, let's say, height. If we're talking about plants, it would be the height of a plant. There might be heritable factors on, let's say, flower color. So flower color. And he recognized that there were different versions of those factors. And so a given plant might have one of the tall versions. So they might have a tall version for the height factor, and they might have a short version. Or they might have two talls, or they might have two shorts, or they might have a red factor, and they have a pink factor. Or they could have two reds, it would look like that, or two pinks would look like that. But the important realization was that there was these versions of the factor. Today, we call these factors, we say, hey, there's a gene for height, if there is one, or that there is a gene for flower colors, and those variations of the genes, today, we call these alleles. So we'd say, hey, you have the variation, you have one copy of the tall allele and one copy of the short. Let me just write it this way. Let me just say these are all alleles right here. So you have one tall allele, one short allele. And what Mendel did is he realized, well, look, these things are he didn't know how, but these things are the things that get passed on from a parent to their offspring. And he started to describe about how they got passed on. He observed that even if you have two of these, that they tend to segregate when you go to the next generation. And what do we mean by segregation? Or I guess we could say the law of segregation. Law of segregation. Well, that means if I'm a pea plant and these are the versions that I have, to my offspring I might pass on an A, a capital A, the tall one, or I could pass on the lowercase a. I might pass on the tall, or I might pass on the red version of the flower color factor, or I might pass on the pink one. And he also realized that whether or not I pass on the capital A or the lowercase a, it's independent of whether I pass on the capital B or the lowercase b. So they independently assort. How this one assorts is independent of how this one assorts. So independent assortment. Independent assortment. Assortment. Law of independent assortment. And he also observed that some of these versions dominate the other one. So if an offspring has a tall version and a short one, if the tall one is dominant, the observed trait will still look tall. And the only way they look short is if they have two versions of the short one. And so that one he described as his law of dominance. Law of dominance. And if all of this is completely new to you, I encourage you to watch the videos on Mendelian genetics on Khan Academy. But this is just an appreciation, a little bit of a historical appreciation. But as big of a deal as Mendel's work was, it's also important to realize what he didn't know. He had no idea of how this was actually happening at a molecular level or at a cellular level. And it wasn't until the early 1900s that people started to have fairly robust theories of how this happens. And so in 1902 and 1903, so 1902, 1903, these two gentlemen independently start coming up with the chromosome theory of inheritance. And it's called the Boveri-Sutton chromosome theory of inheritance because right around the same time, they both started to realize that maybe chromosomes were the actual molecular mechanism, the cellular mechanism by which these factors segregate and independently assort. And so this is, let me write this down. This is the Boveri-Sutton, and sometimes called the Sutton-Boveri, Boveri-Sutton chromosome theory. Chromosome theory. And even though they're starting to say maybe chromosomes have something to do with it, they still don't know exactly what is inside the chromosomes that are allowing somehow this information to be encoded. And we'll get to that. And we will get to that in a little bit. But let me just underline this. Boveri-Sutton chromosome theory. And so what was their key insight? Well, they started to look inside of cells. Meiosis was observed actually after Mendel published his laws of inheritance. And then chromosomes, or how chromosomes behave in meiosis, were discovered after that. And then these guys, they independently studied different organisms. Walter Sutton, he studied grasshoppers. Theodore Boveri, he studied sea urchins. But they looked at meiosis, and they looked at the reproduction and the fertilization during these processes. And they saw that the chromosomes seemed to do things that were very similar to these laws of segregation, laws of independent assortment, laws of dominance. And actually, the law of dominance we'll talk more about in future videos. But he saw that, let's say that you had an organism here, and in this particular organism, I just did it for simplification, it has two pairs of homologous chromosomes. So what does homologous chromosomes mean? Well, these two are different chromosomes, but they seem to be very similar. It seems like they're kind of the same length, same size, same shape. So that's one pair of homologous chromosomes. That's another pair of homologous chromosomes. So notice, homologous chromosomes, two things that are kind of looking the same, but maybe they're a little bit different. We're not sure. Well, maybe this is what fits what's going on right over here with these factors. Maybe, just maybe one of these chromosomes somehow has on it someplace what encodes for the capital A, and maybe the other chromosome in a similar part of the chromosome, has what encodes, the similar part of the chromosome has what encodes for a lowercase a. Now this is starting to make sense because they would be homologous chromosomes, similar. The chromosomes look like they code for the same thing, for the same factors, for the same genes, but there might be some variation between these chromosomes. And these guys weren't, they weren't able to somehow sequence the chromosomes, so they didn't know. They didn't even know that the DNA was what was important in the chromosomes. But they said, well, it looks like these two things, if we look through the process of meiosis, it seems like they segregate from each other. For example, this capital A one, it'll replicate. So you have capital A, and then this is the lowercase a one right over here. You might have some crossover, and we'll talk about that when you could review meiosis if that looks unfamiliar. But then they segregate. You could have your capital A ones right over here, and then these sister chromatids split apart. So capital A, and then you have lowercase a. These lowercase a ones, they segregate. And they independently assort from the other chromosomes. So this one right over here might be the capital B. This might be the lowercase b. And whether or not this gets a capital B or a lowercase b is independent of whether it got a capital A or a lowercase a. So it seems like these chromosomes independently assort. And so they came up with this chromosomal theory that it looks like maybe chromosomes are what contain these heritable factors that Mendel was talking about, because it seems like chromosomes behave very similar to those heritable factors. Maybe chromosomes code for multiple of these heritable factors that segregate and independently assort. And as we know now, they were right. So this was a very, very big deal. But it's important to realize that they weren't sure. They established the theory. They were able to make some observations with the grasshoppers and the sea urchins, and they saw the patterns between what Mendel was describing and the way chromosomes behave during meiosis. And then they know that each of these products of meiosis, each of these gametes will then go and form with other gametes to form the next organism. So you say, oh, look, parents will contribute either a capital A or a lowercase a, either a capital B or a lowercase b. So it says this is very similar to what Mendel was describing. So they laid the groundwork for the theory, but they still weren't sure. They didn't definitively prove it. And it will take another decade or so until it's definitively proven. And even then, no one was really sure exactly how these different traits were encoded. And for that, we would have to wait a little bit longer.
So folks, this work, the work of Sutton, the work of Bovieri, tied together the ideas of Mendel with a mappable and observable thing that Sutton and Bovieri saw. Mendel saw very clearly about the idea of pairing, the idea of segregation, the idea of independent assortment. All of this is verified by simply examining and mapping the pattern by which chromosomes moved. So from the above observations, from what Sutton and Bovieri saw in relation to the pattern of chromosomal movement, they formed what we now know as being the chromosomal theory of inheritance, where it states that Mendel's factors are indeed carried on chromosomes, and that the segregation and independent assortment of chromosomes during meiosis accounts for the pattern of inheritance in an organism. It is the chromosomal movement that accounts for how traits are passed from parents to offspring. Now, there was some debate with that, but what was really copper fastened, if you will, nailed down, if you will, the idea of chromosomes being where these traits are found, was the work of an American scientist by the name of Thomas Hunt Morgan. And he actually worked with fruit flies, and he saw patterns that he observed there. Specifically, let's take a look at what he saw.
Professor Dave here. Let's check out some genes.
He knows a lot about the science stuff. Professor Dave explains.
As we already know, Mendel described factors of inheritance called genes, but he did this before anyone knew about the existence of the chromosomes that contain them, or the DNA they are made of. The early 20th century saw incredible progress in contextualizing Mendel's work within new discoveries in molecular biology, as it was then possible to see chromosomes in a microscope and watch them replicate and redistribute themselves during cell division, which led to the chromosome theory of inheritance. It was at this point that studies regarding mitosis and meiosis were able to explain Mendel's laws of segregation and independent assortment, as we realized that the two alleles for a gene separate during gamete formation, and that alleles for two different genes separate in random combinations, so long as they are on different chromosomes, as the chromosomes themselves, which contain the genes at specific loci, will undergo segregation and independent assortment during gamete formation. But more evidence was still needed to corroborate the chromosome theory of inheritance, so let's see what happened next. Some very compelling evidence for the chromosome theory of inheritance came from the work of Thomas Hunt Morgan. He studied fruit flies, which have only 4 pairs of chromosomes, those being 3 pairs of autosomes and 1 pair of sex chromosomes, or an X and Y chromosome, just like humans. 2 X's for a female and an X and a Y for a male. In studying the flies, he noticed a rare characteristic that sometimes appeared in males, and that was white eyes instead of the typical red. If considering phenotypes, red eyes would be referred to as the wild type, and white eyes would be considered the mutant or non-wild type. Morgan did some experiments that were similar to Mendel and the pea plants. He bred a red-eyed female with a white-eyed male. The entire F1 generation had red eyes, suggesting that red eyes are dominant, which is consistent with Mendelian genetics. Then he bred the F1 generation amongst itself, and he observed the expected 3-to-1 ratio for red to white eye color in the F2 generation, with an additional result. White eyes showed up only in males. All the F2 females had red eyes, and half the F2 males had red, while half had white. This was firm evidence that eye color was linked to sex, which makes perfect sense considering the discrepancy in sex chromosomes for males and females. It was suggested that the gene for eye color in fruit flies was an X-linked gene. The reasoning went as follows. The original parental generation was a red-eyed female with 2 X chromosomes, each with the red allele, which is represented by the letter W with a plus in superscript, and a white-eyed male with an X chromosome containing the white allele, represented by the letter W, and a Y chromosome, which does not contain the gene in question. In producing gametes, all the female eggs would have an X chromosome with the red allele, while for the male, half would have an X chromosome with the white allele and half would have the Y chromosome without this gene. This means that all of the F1 generation would have precisely 1 red allele and thus red eyes, since this is the dominant allele. But then, for the F2 generation, 25% would be homozygous dominant, 50% would be heterozygous, with the presence of the white allele or the Y chromosome being irrelevant to the phenotype, and 25% would have the white allele and the Y chromosome, thus missing the red allele and possessing white eyes. But all of these flies will necessarily be male, which is indeed what was observed. This constituted tremendous evidence for the chromosome theory of inheritance, because it demonstrated that information regarding the inheritance of a particular phenotype was associated with a particular chromosome, in this case, the X chromosome. Beyond corroborating the chromosome theory of inheritance, it was therefore realized that sex-linked genes have interesting patterns of inheritance. It is worth noting that not all organisms that reproduce sexually have the same XY system of sex determination that humans and flies do, which we learned about when we studied meiosis. Instead, some other systems are as follows. The X0 system is found in certain insects, like grasshoppers and cockroaches. Here, there is only one sex chromosome, the X chromosome. Females have 2, and males have 1. Their sperm may contain an X chromosome or no sex chromosome. In various birds, fishes, and certain insects, a ZW system is utilized. Females are ZW and males are ZZ. A bit stranger is the haplodiploid system found with bees and ants. Here, there is no sex chromosome. It is simply that eggs that are unfertilized develop into males, while eggs that are fertilized develop into females. This means that male bees and ants, having no fathers, are haploid organisms. All of their cells have only one set of chromosomes, just like our gametes, while females are diploid, each cell containing 2 sets of chromosomes, just like most other animals. With sex-linked genes better understood, we can examine the inheritance of certain traits like color blindness, which is an X-linked disorder. The normal allele is represented by the letter X with a capital N in superscript, while the recessive allele has a lowercase n in superscript. Say that a homozygous dominant female reproduces with a colorblind male, meaning one recessive allele and a Y chromosome, lacking the gene entirely. Offspring have a 50% probability of being female and heterozygous, and 50% probability of being male with the dominant allele, a situation we would call hemizygous dominant, where hemizygous means that only one allele is present. Thus, there is a 0 probability of passing on color blindness, but a 50% probability of producing a carrier. Now, if the mother is heterozygous and the father has the dominant allele, there is a 25% probability of offspring being homozygous dominant, 25% heterozygous and therefore a carrier, 25% hemizygous dominant, and 25% hemizygous recessive, and therefore colorblind. Lastly, if the mother is a carrier and the father is colorblind, there is a 25% chance offspring will be a carrier, 25% hemizygous dominant, and a 50% chance of being colorblind, with the possibility of producing a colorblind female, which is far more rare, given that both parents must possess the rare recessive gene. There are a large number of X-linked disorders, given that the X chromosome is so much larger than the Y chromosome, containing some 1,100 genes, whereas there are far fewer Y-linked disorders since it has only 78 genes, some of which are duplicates, although these can exist as well. Now that we understand X-linked genes, let's move forward and look at gene linkage in a more general sense. Thanks for watching. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.
So again, that's the chromosomal theory. We've looked at that. And the work of Thomas Hunt Morgan really crystallized the notion that it was the chromosomes and how they were passed on that really accounted for trait transmission. And again, just to recapture there, we know that in fruit flies, normally the dominant or wild color is red eyes. Flies would normally have those red eyes. But what he did is he took 2 red-eyed parent flies, crossed it, and they got a white-eyed male. Red-eyed females, red-eyed males, white-eyed males. Because there was that linkage that the possession of the white color seemed to be something related to the sex, he concluded that eye color was linked to an organism's sex. Now, because it was known that sex was linked to the chromosome, if eye color is linked to sex and sex is linked to a chromosome, it's a natural conclusion that eye color is linked to a chromosome. So if you look at what he did, again, using a Punnett square, he hypothesized that the chromosome must be carried on the X chromosome. Now, we know that fruit flies have XY just like humans. So if the gene for eye color is only carried on the sex chromosome, and we cross this, we would see a red-eyed male cross with a red-eyed female, and you would get this: X big R, X little r, X big R, X little r. If they're heterozygous. Or looked at another way, let's take a look and break this down. So know that in fruit flies, red is dominant to white. So we have two red-eyed flies, X bigger, X bigger, cross with X big R Y. Of course, you would have a pure line. Okay, all well and good. Which means that you would always and only get red eyes. All pure lines. However, what he did find in one of his crosses is that he found that I got a white-eyed fly. Where'd this come from? He hypothesized that what likely occurred is that you had a female that was heterozygous. So taking that for this sex-linked trait, meaning carried on the X chromosome. So if the female was heterozygous and the male was still red-eyed, phenotypically red eyes. Okay, X bigger, X bigger, X bigger, Y. X bigger, X little r, X little r, Y. You got the 3-to-1 ratio as expected by heterozygous. Okay, all well and good. However, there was this linkage towards sex. So then what he did is he took this white-eyed male, and he crossed it. He crossed the white-eyed male, which would be X little ry, with a heterozygous red-eyed female. When you do those crosses, let's see what we get here. X little r, Y, X big R, X little r. You could indeed get a white-eyed female. But the only way you could get a white-eyed female is if the male was himself white-eyed. So this linkage between sex and the chromosomes, this relationship between the trait and sex, gave credence to the notion that there was a linkage between chromosomes and traits. So if you look at what Morgan discovered, he discovered that there was a linkage between chromosomes and sex. But that's true for all traits. But it gave credence to the notion that's exactly what it was. He also stated that genes that are located on the same chromosomes would be linked to each other, and they wouldn't sort independent. Why? You're getting a package of alleles, which is why the process of crossing over, as we looked at, is so important. It helps create recombinants, and these are called linked genes, genes that are found on the same chromosomes. So if you look at sex-linked chromosomes, we know that there's a transfer on the X or Y from one generation to next. Genes only located in the X chromosome are sex-linked. Now, if there are the Y, they're Y-linked, which means females wouldn't have it. Now, the reality is there's very few Y-linked traits in humans, mainly because the Y chromosome is very small. It doesn't have a lot of space for genes. I guess one thing that might be there is pattern baldness, for example. Now, what he did find is that there were some genes that do segregate. In other words, these we found on different chromosomes. Now, he formulated the gene chromosome theory, which states that genes exist at specific sites and are arranged in a linear fashion along chromosomes. Now, genes are located from one end of the chromosome to the other, as you see in this illustration. Now, if they're linked together, they're going to be inherited together, resulting in less variation. But the process of crossing over can provide a shuffling of the deck to actually provide enhanced variation as chromosomes exchange sections and shuffle things up a little bit more. Or let's take a look once again, folks.
Place. Now, crossing over takes place during meiosis, and that means crossing over occurs when we form gametes, sex cells. Now, what exactly is the purpose? What's the importance of crossing over? Well, as we'll see in just a moment, crossing over allows us to produce sex cells. It allows us to produce gametes that carry genetic information that is slightly different than genetic information found inside our somatic cells. And that's exactly why I, for example, don't look exactly like either of my parents. And that's exactly why I have my own set of unique fingerprints. It's because crossing over allows those gametes to obtain genetic information that is unique to that specific sex cell. Now, in our discussion of crossing over, we're going to focus on a type of organism we call the fruit fly. Now, in fruit flies, we're going to study two different traits. We're going to examine the structure of the wings, and we're going to discuss the color trait of that fruit fly. Now, in fruit flies, we have two types of wings that can form. We have normal wings and we have vestigial wings. Likewise, we have two different types of colors. We have the gray color and we have black color. Now, it turns out that normal wings that we're going to designate with blue uppercase V is dominant over the recessive vestigial wing, which we're going to designate with purple lowercase v. And by the same token, the color gray is dominant over the color black. So the color gray we're going to designate with red uppercase B, and the color black we're going to designate with orange lowercase b. Now, the normal wings and our color, or the wing gene and the color gene, are linked to one another. And what that basically means is they are found on the same exact chromosome. So to see what we mean by that, let's take a look at the following diagram. So this is basically a homologous chromosome pair. And what that basically means is one of these chromosomes came from the male parent and the other chromosome came from the female parent. And both of these chromosomes carry genes that code for the protein that expresses the same type of trait. Now notice along each one of these chromosomes, let's suppose along the darker green chromosome, we have two genes. And this red gene is linked to the blue gene in the same exact way that this orange gene is linked to the purple gene. So whenever two or more genes are found on the same chromosome, they are said to be linked to one another. So this gene right here, uppercase V, is linked to this here, uppercase B, while this is linked to this right over here. Now, let's suppose that this particular fruit fly that has this homologous chromosome pair wants to produce gametes. So it wants to undergo reproduction, and that means it wants to produce gametes. And the first step in producing gametes is to replicate the DNA during interphase. So once we replicate the DNA, we form the following two homologous chromosomes. So now instead of having a single chromatid, we have two sister chromatids. So these two, so this is a sister chromatid to this. And because DNA replication produces identical pieces of chromosome, that means these two are identical chromosomes. By the same exact analogy, this is identical to this. So these two are sister chromatids with respect to one another. So these two are sister chromatids, these two are sister chromatids. Now, these, with respect to one another, are homologous chromosomes in the same way that these two were homologous chromosomes up here. But now, once DNA replication took place, we form these two identical pairs of sister chromatids. So these are identical to one another, these are identical to one another, but these are different with respect to one another. But we call them homologous because they still carry those genes that code for the same exact traits. So we have the wing structure and the color, and the wing structure and the color traits. So we know that during meiosis 1, in our discussion on meiosis 1, we said that during prophase 1 of meiosis, we have a process known as synapses taking place. So the synapses is basically the intertwining of the chromosomes. And when the chromosomes intertwine, when they overlap onto one another, we have crossing over taking place. And what crossing over is, it's the exchange of genetic information, of chromosomal pieces among two non-sister chromatids. So remember, these two are sister chromatids with respect to one another. These two are sister chromatids with respect to one another. But this one here and this one here are non-sister chromatids. That's because they are not identical to one another. Now, when crossing over actually takes place, we form genetically different chromosomes. We form recombinant chromosomes. So once again, during meiosis 1, synapses between the homologous non-sister chromatids, so let's say this one right here and this one right here takes place, and that allows the exchange of genetic information in a process known as crossing over, and that produces genetically recombinant chromosomes. So basically, during prophase 1 of meiosis, these two homologous chromosomes that have been replicated basically approach one another. And now this right here can basically overlap with this right here, and when this interlapping or overlapping takes place, this is known as synapses. So we have this overlaps onto this right over here. Now, when the overlapping takes place, then we can exchange in the process known as crossing over. And this dark green piece that came from this here goes onto the light green piece onto this chromatid right over here. And likewise, this lighter green section of the chromosome that came from this homologous chromosome goes on to this. At the end, we form the following pair of homologous chromosomes, in which this is no longer identical to this, and this is no longer identical to this. And at this point, we call these recombinant chromatids. Why? Well, because none of these are actually identical, as in the case of this, where this one was identical to this and this one was identical to this. Now, each one of these are genetically unique. So this is basically what happens during prophase 1 of meiosis. Now, let's fast forward to metaphase 1 of meiosis. During metaphase 1 of meiosis, these recombinant chromosomes basically align along the equator, along the midsection of that particular somatic cell. So this is the midsection, and this homologous but recombinant chromosome pair have now aligned itself along the equator, and now a process known as segregation will take place. So these will basically separate onto opposite poles. Eventually, cytokinesis will produce two different cells. And this is the reduction step, where we go from the 2n number to the n number of chromosomes. Now, let's suppose now we fast forward to metaphase 2 of meiosis. During metaphase 2 of meiosis, each one of these individual chromosomes will basically now align itself once again along the equator, and the same segregation process will take place. So the mitotic spindle will attach itself onto these chromosomes, pulling them apart, and eventually we form these 4 gametypes as shown in the following diagram. Now notice in each one of these gametes, we have a slightly different genetic information, and that is because of the process of crossing over. So when crossing over actually took place, we exchanged genetic information. Notice that in this case, we had uppercase V, uppercase B, and uppercase V, uppercase B. Okay, but when genetic recombination, when crossing over took place, we have uppercase V, uppercase B, uppercase V, and now we have a lowercase b. And so that's exactly what makes this one different than this chromosome right over here. And that's exactly why we produce these 2 gametes. So this gamete is genetically different than this gamete. Now, if no crossing over actually took place, then these 2 gametes would be identical because this piece of information would never have been exchanged in the first place. And if no crossing over took place, this would be identical to this. By the same exact reasoning, if we examine this cell right over here, when these separate to opposite poles, cytokinesis takes place, we form these 2 gametes, and these are genetically different from one another because of this process of crossing over. So in this case, we had lowercase v, lowercase b, lowercase v, lowercase b, and these were identical sister chromatids. But now, after crossing over, this right over here is lowercase v, but this is uppercase b because of crossing over. Now, this is still lowercase b and lowercase v, but this chromatid here is no longer identical to this chromatid here. And so that means these 2 will be different, and so when they slightly different gametes. So this gamete will contain a slightly different genetic information, slightly different chromosome than this one right here. But if no crossing over took place, we would form 2 identical chromosomes, lowercase b, lowercase v, and lowercase b, lowercase v. Instead, because we do have crossing over that takes place, we form one lowercase b, one lowercase v, and the other one is uppercase b, lowercase v. So this is exactly what we mean by the process of crossing over. So we see that crossing over allows diploid organisms such as human beings to basically produce these sex cells, these gametes, that have slightly different genetic information than the actual parent, than the parent or than the female or the male parent. Because remember, this chromosome came from the male parent and this chromosome came from the female parent. If no genetic recombination in the form of crossing over took place, then the gametes that we would form at the end, these gametes here, would be exactly identical to the gametes that came from the parents, the male and the female. But because of crossing over, we form these gametes that are slightly different, and that means we're going to form individuals. Our offspring will be genetically unique and genetically different than our own somatic cells.
So in essence, what happened when you start drilling down, looking at, you get unexpected results. You get recombinants that you wouldn't necessarily expect. To that end, this gave crazy idea, well, if the genes are linked, how do we get these? Well, we knew that they saw the notion that you would get recombinants forming. Remember, chromosomes sometimes undergo crossing over. They exchange sections during meiosis. When this happens, a segment of DNA on one homologous chromosome will actually exchange a section with the other. So this crossing over, you get that this exchange of sections between homologs. As a result, you enhance or recombine the sets of genes. This occurs in meiosis. We looked at previously, the process synapsis. Now, what ends up happening is you get additional combination resulting, which could have additional possibilities forming amongst gamete makeup. So you see Morgan's data right here, where things were actually established. What Morgan derived, what Morgan looked at, he saw the way that these recombinants actually formed. Again, this is all directly from your textbook. So this allowed Morgan to find the ability to actually map chromosomes. Where are chromosomes located to? Where are they linked? Remember, the linkage groups that correspond respond differently. Now, if genes were always found at the same location on the chromosome, now, if it wasn't true, you wouldn't get any exact exchange of alleles, but you did. Now, Morgan's experience showed that the frequency of crossovers between any two links was always the same. This allowed him to generate data, which allowed him to formulate the notion of recombinant maps by getting a crossover percentage. In other words, what is the total number of recombinants divided by the total number? So looking at an example, the crossover percentage in an offspring in table 2 that we just put up was 4.5%. 18 recombinants out of a total of 400. This allowed a generation of that data. So when you're recombining things and you see where they're located to, this percentage of recombinants allowed it to map out where they would do in relation to each other. So we're done as such.
Hi, it's Mr. Anderson, and in this video I'm going to talk about genetic recombination and gene mapping, and it centers on the work of Thomas Hunt Morgan, who used fruit flies to show that genes just don't travel by themselves. They actually travel on chromosomes, and as those chromosomes undergo what's called crossing over, genes from one chromosome are actually going to swap position with genes from another chromosome. And so before we get to that, we should talk about fruit fly genetics for just a second. And so on the left we have a wild fruit fly. That's what they normally look like. And on the right is a mutant. There are two mutations in the one on the right. Not only coloration, but you can see that it also has these really small, what are called vestigial wings. And so if we look at the genotypes, the one on the right is little b, so it has that black coloration. The one on the left, we simply add a plus to it, and that implies that it's of the wild type. We could also look at vestigial wings. Maybe the genotype of the one on the left has one of the wild type normal wings, but it has one of the vestigial genes. It still has normal wings on the left. And that's because the wild type is going to be dominant in this case. And so let me show you the quintessential cross that Morgan did that was so puzzling. And so what he has is a normal wild type on the left, but it's hybrid for both of these genes. And so you can think of this as like the F1 generation, and then he's simply doing a test cross with it. So he's crossing it with a mutant fly that's mutant and homozygous recessive for both of those traits. And so Morgan understood the work of Mendel, and so he set up his Punnett square like this. And so on the top, he's going to show all the possible gametes that we could get from this one parent. So you could have both of the wild type genes, or you could have both of those recessive mutant genes, or you could have a combination of the two. So you could have one wild, one recessive, or vice versa. Now this parent over here only can give its recessive genes, and so we could represent that on the other side like this. And so we knew that there are only 4 possibilities that we could get out of this. And those first 2 are going to look like that. And we call those what are called the parental phenotypes. Why is that? Because this one looks like that parent, and this one looks like that parent. In other words, there's no recombination. But on these other two alternatives right here and right here, what we're getting is a recombination of those parents, and so we call these simply the recombinant phenotypes. But that shouldn't have been confusing to him. If we look at the Punnett square, we have 4 different squares, and so we would expect that 50% are going to be parental and 50% are going to be recombinants. But when he did this cross, what he found is that there were actually 17% recombinants and 83% that were of the parental type. And so was Mendel wrong? Was all of this wrong? No, it's just that the model wasn't good enough. And so he thought about this idea of 17% and what it meant for a really, really long time. And then finally, one of his students, Alfred Sturtevant, and I couldn't find a good open source picture of him, but he's always smoking a pipe. So we'll say this represents Alfred Sturtevant. One night just blows off his homework and he figures it out. The whole thing, he figures it out. And to understand it, you really have to understand what's going on during meiosis. And so if we look at these two parents, so this is the double mutant on this side, and this is the hybrid on the other side. Let's look at each of those and figure out what gametes could they produce. And so if we look at the one on the right, we know that it can only produce these 2 gametes, but since we're seeing a frequency of recombination that's less than 50%, that implies that these 2 genes are found on the same chromosome. We know this now. Thomas Hunt Morgan and Alfred Sturtevant had to kind of work through this. But if we look at what does that mean, these 2 genes are found on the same exact chromosome. So if we go through all the steps of meiosis, remember what happens first during interphase is that we copy all of the DNA, and then it divides in half, and then it divides in half again. And since those genes were on the same chromosome, I see just one possible gamete that could be produced. In other words, you're going to get one of each of those recessive genes. Now let's look at that hybrid parent. And we know, and Thomas Hunt Morgan knew, since he saw some of those recombinants, we had to have all 4 of these possible gametes. And so let's put the dominant or the wild type genes on this one chromosome and the recessive on another. So how do I know that I have both of the wild type on one chromosome and both of the recessive on another? Remember, this is the F1 generation, and so it's receiving this chromosome from a parent that was pure for both of these genes and vice versa for the pure mutant parent as well. And so let's go through the steps of meiosis again. And so what happens during interphase is that we copy them, then there's one division, and then there's another division. And so how many gametes do you see? Well, this one is exactly the same as that one. And it's not based on orientation of the chromosomes because, again, they're both found on the same chromosome. And so this was puzzling, but then eventually they settled on this idea of crossing over. What if there were crossing over between these chromosomes? What if somehow this chromosome wrapped around this chromosome during meiosis, and they could see that under the microscope, they could see this occurring. If these crossed over, what you could get is bits of this chromosome actually being crossed over to that one. And so what we could now produce is a chromosome that has the wild type for coloration, but it has the recessive gene for this vestigial wing, and vice versa over here. And so Sturtevant, it's brilliant coming to this kind of idea that if that crossing over event occurs between the different genes, then we would have recombination, genetic recombination. But if it doesn't occur during that, in that part of the chromosome, there's going to be no recombination. And so where does that 17% come from? Well, this is roughly 17% of that area of the chromosome. That's where it's coming from. If those genes were closer together, that frequency recombination would be closer. If they were really far apart, it's more likely that it's going to split in the middle. And so we can use this one cross to figure out the frequency of recombination, and then they were able to use that to build a gene map. And so if you look at a chromosome, if we look at that frequency of recombination, let's say it's 17%, that implies that it's an arbitrary distance of 17 map units apart on the chromosome. Let's say the frequency of recombination is less than that. That means the genes are closer together. What if the frequency of recombination is greater than that? It means that it's farther apart. What if it's exactly 50%? Remember, that's what we were thinking about. If it was independent assortment, that would mean that those two genes are found on different chromosomes. And so we can use that to really map a chromosome. And so let's look at some of the data that they gathered. They found that the distance between the vestigial and that black coloration gene, the frequency of recombination is 17%. They then compared that to another gene called the cinnabar, which has to do with the eye coloration of the fruit fly, and they got these frequency of recombinations as well. And so when you're figuring out a gene map, what I would encourage you to do is always start with the highest frequency of recombination. So I'm going to start with this one and just choose to put them on that chromosome, we'll say 17 units apart. So we're going to put the vestigial and the black apart by 17. Now let's go to another one. So let's figure out where these cinnabars are. Well, if we start with the vestigial gene, we know it's going to be 8 map units apart from that. So I could say maybe it's going to be over here, or I could say it's going to be over here. So we have these two different alternatives. And so which of those actually fits with that last frequency of recombination? Well, if I put it way over here, then we're going to have a frequency of recombination between that and the black. We know it to be 9%, but it's going to be a way larger number than 9%. And so I can narrow it down to this is where our gene map fits. Now let me give you a problem of your own. So now I've given you these 4 genes and their frequency of recombination. I would encourage you to pause the video here and then you try to map out where each of those genes are found on the chromosome. I'll pause, and then let me show you what the right answer is. And so what I would do is, again, start with the largest frequency of recombination. I'm going to put B and C really far apart. So I'll put B on one side, C on the other. What's the total distance of the chromosome? Remember, it's going to be 50 map units. And now I can work backwards. And so now let me figure out, so I've got B and C. Where's D going to be? Well, I can't put D way out here because I don't have enough map units to do that. So I'm going to have to put it over here. And once I've got D, I got to figure out where A is. So I could work backwards to that. Well, I know that A can't be way out here on this side, so I know that A's got to be somewhere over here. So that would be the relative map distance, or the relative gene map, based on frequency of recombination. And so Sturtevant and Morgan did that over years, and they were able to map out where the genes are found on the chromosomes. Now, we don't do it this way anymore. What do we do today? We simply sequence the DNA. Once we sequence the DNA, we can figure out where the genes are. But the cool thing is that as we compare that, you could go right here to the fly base. I was looking up the vestigial gene. We know exactly where it is, but that maps up perfectly with the work of Morgan and Sturtevant. And so that's genetic recombination. It allows us to create gene maps, and I hope that was helpful.
So again, expanding on this work here, you can see how we work through this exercise here. For example, from cross between different fruit flies, Drosophila, geneticist finds that the crossover frequency between A and B is 12%, between B and C is 7%, and between A and C is 8. Or 5 rather. So we look at these numbers. If gene A was in the middle, then the sum of the distance between B and A and C must be the same. So B to A is 12. If it was A to C was 5. So therefore B to C would have had to be 7. Now if you add them up, 12 plus 5 does not equal 7. So if gene B was in the middle, then the sum of the distance between A and B and between B and C, again, would have to equal the distance between A and C. These aren't equal, so it's not in the middle. As we see here, if A and B with C in the middle and B and C is 7, well, again, doesn't equal. But if we put gene C in the middle, the sum of the distance between A and C and C and B would have to equal this between A and B. Between A and B is 12. Between B and C is 7. 7 plus 5 is 12. Therefore, the sequence would have had to be A, C, B. So, in looking at chromosome theory of inheritance states that chromosomes carry the genes. Each chromosome contains many different genes, the pair of chromosomes. All these things play through. So thanks for your attention, everyone. We'll see you in class on Monday. Make sure you watch this material. Make sure you stay along.